Welcome back to another video, but this one is not the usual paper 1-2 series. Today I'll be going through the 9700 October-November 2019 paper 1-1. If you are new to this channel, join the DF Edu squad by clicking the subscribe button. If not, well, do continue watching. Also do hit that like button, and without wasting further more time, let's get started. Question 1. Which cell structure contains cytoplasm? A. Chloroplasts, B. Mitochondria, C. Plasmodesmata, D. Smooth endoplasmic reticulum. Now, the fluid in mitochondria is not the cytoplasm, but it is called the matrix. The fluid in chloroplasts is called stroma, and SER is present in the cytoplasm but do not contain cytoplasm. Hence, that leaves us with the answer being C. Plasmodesmata. If you recall the definition of plasmodesmata, plant cells are linked to neighboring cells by means of fine strands of cytoplasm called plasmodesmata. Hence, it does contain cytoplasm. Therefore, the answer is C. Question 2. Some cell structures are listed in a particular order. 1. Nucleus, 2. Ribosome, 3. Golgi body, 4. Vesicle. What determines the order in which these cell structures are listed? If you look at cell structure 4, vesicle, vesicle formation is involved in either exocytosis or endocytosis. Therefore, if we go to the options A, B, C and D, option B talks about the synthesis of an antibody, and antibody formation does involve exocytosis, as antibody is a protein and the sequence does show the production of a protein due to the transcription process occurring in the nucleus, followed by translation process occurring in the ribosome, and then the modification of the protein, and finally vesicle formation in order to secrete the antibody out of the cell. Hence, the best possible answer is option B, sequence used in the synthesis of an antibody, as this sequence talks about the production of a protein. Question 3. The drawing has been made from a section showing part of an alveolus and a red blood cell in a capillary. The magnification of the drawing is times 5000. What is the minimum distance that oxygen must diffuse from air in an alveolus into the red blood cell? This question right here involves calculation. Using the formula, magnification is equal to image size upon the actual size. Since they are talking about the minimum distance, you have to place your ruler and measure from the alveolus into the red blood cell, but where the distance is the smallest. Hence, if you calculate using the magnification given, that is times 5000, and the measurement you obtain, that is the image size, you will get the actual size as 1 micrometer. Hence, the answer is D. Question 4. 4 students were asked to match the function with the appearance of some cell structures in an animal cell. The functions were listed by a number. 1. mRNA passes through the ribosome. 2. Synthesis of polypeptides. 3. Packaging of hydrolytic enzymes that will remain in the cell. The appearances were listed by a letter. V. Membranes which surround an enclosed inner cavity. W. Non-membrane bound spherical structures. X. A double membrane interspersed with pores. Y. Non-membrane bound cylindrical structures. Z. Membrane bound sacs arranged as a flattened stack. Which student correctly matched the numbered functions with the appearance of the cell structure? Now, the appearance V describes the endoplasmic reticulum. Appearance W describes the ribosome, as ribosomes have no membranes. X, since it's a double membrane with pores, it should be describing the nucleus. Y, non-membrane bound cylindrical structures, that describes the centriole. Z, membrane bound sacs arranged as a flattened stack, that is certainly the Golgi body. Therefore, if we match the correct function to the appearance, you get 1. mRNA passes through to the ribosome, being transcription process which occurs in the nucleus. Hence, the appearance should be X, as X talks about the nucleus. Therefore, 1 matches with X. That leaves us with options being C or D. If you look at function 2, synthesis of polypeptides, that certainly occurs in the ribosome. Hence, should be matching with the appearance W. Therefore, that leaves us with answer C. Question 5. The antibiotic inhibits protein synthesis in mitochondria and in some prokaryotes. The antibiotic does not inhibit protein synthesis in the cytoplasm of eukaryotic cells. What would be the effect on the cells of a person being treated with the antibiotic? I have highlighted the key part of this question: inhibits protein synthesis in mitochondria. Now, mitochondria is an organelle that is involved in ATP production, which releases energy. Hence, if it inhibits the protein synthesis in mitochondria, then the mitochondria which is involved in ATP production, the rate of ATP production will decrease. That leaves us with the answers being A or B. With decreased rate of ATP production, there would be reduced activation of RNA nucleotides, resulting in reduced transcription of nuclear DNA. As activation of RNA nucleotides requires energy, and with reduced ATP production or reduced energy production, that leaves us with the activation of RNA nucleotides being less, and activation of RNA nucleotides is involved in the transcription process. Hence, transcription will also decrease. Therefore, the answer is A. Question 6. The diagram shows the monosaccharide xylose. Many xylose molecules can be joined by glycosidic bonds to form a polysaccharide which is found in plant cell walls. Which diagram shows the formation of a glycosidic bond between the two xylose monomers? Now, glycosidic bond is formed between two OH groups, and it is formed by the process of condensation. And condensation is nothing but when a water molecule is removed, a bond is formed. Hence, if we go to the options A, B, C and D, the glycosidic bond should form between two OH groups, and that leaves us with the only answer being D. Therefore, D is the correct answer. Question 7. Which color indicates the lowest concentration of reducing sugar present in a solution after testing with Benedict solution? Now, when you test a substance with Benedict solution, the color scale is from blue to green, green to yellow, yellow to orange brown, and then last to red. Red being the highest sugar concentration and blue being no sugar concentration. Since they are asking about the lowest concentration of reducing sugar present, it should lie towards the left end of the color scale, that is from blue. Hence, if we go to the options A, B, C and D, C is obviously incorrect as red leads to higher sugar concentration. D is also incorrect. A is also incorrect as that is also on the right hand end of the color scale. Therefore, the correct answer is green. Answer B. Question 8. Which molecules are monomers? 1 ribose, 2 glucose, 3 deoxyribose, 4 sucrose. If you look at option 4, sucrose. Sucrose is a disaccharide and it is made up of fructose and glucose. Therefore, it is not a monomer. And since answers B, C and D contain option 4, that leaves us with A being the right answer. 1, 2 and 3 only. Question 9. Which molecules have properties that are dependent on hydrogen bonds? 1 cellulose, 2 glycogen, 3 hemoglobin, 4 water. Glycogen has only glycosidic bonds and there are no hydrogen bonds involved anywhere in glycogen. Therefore, option 2 is obviously incorrect. And if you go to the answers A, B, C and D, A, B and D has option 2 in it. Hence, that leaves us with the correct answer being C, 1, 3 and 4 only. The others all have hydrogen bonds except glycogen. Question 10. Some features of triglycerides are listed. 1 can be liquid or solid at room temperature. 2 contains a high proportion of carbon-hydrogen bonds. 3 insoluble in water. 4 less dense than water. Which of these features makes triglycerides suitable energy stores? Good energy stores are those which are insoluble in water, as it does not affect the osmotic properties, and it should also have many carbon-hydrogen bonds. Therefore, if you go to the options, 2 contains a high proportion of carbon-hydrogen bonds, yes, that is correct. 3 insoluble in water, yes, and that is why energy can be stored as a reserve. Therefore, the answer is D, 2 and 3 only. The others do not contribute in making it a good energy store. Question 11. Some foods contain hydrogenated vegetable oils. These are unsaturated fats that have been converted to saturated fats. Which property of the fats will have changed? Now remember, unsaturated means double bonds are present. Saturated means no double bonds are present. Since it is changing from unsaturated fats to saturated, that means double bonds will be removed. And a key thing is, unsaturated fatty acids, they have bent tails, as when there is a double bond that leads to a kink or a bent tail, whereas saturated doesn't have bent tails, therefore will pack more closely together as it has no double bonds. Therefore, the answer is A. Their hydrocarbon chains will fit together more closely. If we go to the options B, C and D, B, their solubility in water will increase. No, that is incorrect, as fluidity is concerned with double bonds being present. Therefore, option B is incorrect, as no double bonds are present. If we go to C, they will have more double bonds. That is obviously incorrect, as saturated fats have no double bonds. Answer D, they will remain liquid at room temperature. That is also incorrect, as only unsaturated fats can be liquid, but saturated fats are mostly solids. Therefore, they won't remain liquid at room temperature. Hence, the answer is A. Question 12. Which statements could be used to describe enzyme molecules and antibody molecules? 1. Hydrogen bonds stabilize the structure of the protein and are important for it to function efficiently. 2. Hydrophilic R groups point into the center of the molecule and causes it to curl into a spherical shape. 3. The tertiary structure of the protein molecule plays an important role in the functioning of the protein. Statement 3 is obviously correct, as tertiary structure does play an important role in the functioning, as it determines the shape of the active site of the enzyme. Therefore, statement 3 is obviously correct. Statement 2, hydrophilic R groups point into the center. That is incorrect, as only hydrophobic R groups point into the center, whereas hydrophilic stands for water loving. Hence, it will lie towards the outside region, pointing to the watery solution or watery surroundings. Therefore, 2 is obviously incorrect. It doesn't point to the center but to the outer surface. Hence, if we go to the options A, B, C and D, A, B and D have statement 2 in it. Hence, they are all incorrect. That leaves us with answer C being the right answer. 1 and 3 only. Question 13. The Vmax and Km are determined for an enzyme catalyzed reaction. What will be the effects on the Vmax and Km in the presence of a competitive inhibitor? They're talking about the presence of a competitive inhibitor, and when in the presence of a competitive inhibitor, the Vmax doesn't change. It is unaffected, whereas the Km will increase as affinity towards the substrate decreases when there's any sort of inhibitor. Hence, if the affinity decreases, the Km increases, as they have the opposite correlation. Since the Vmax stays the same, that leaves us with answers being C or D, and since the Km increases instead of decreasing as the affinity decreases, that will leave us with answer being D. And remember, Vmax is only lowered when in the presence of a non-competitive inhibitor. If there is a competitive inhibitor, the Vmax is unchanged. Question 14. The graph shows how the concentration of components 1, 2, 3 and 4 of an enzyme catalyzed reaction changes with time. Which row identifies the components of this reaction? With time, obviously the concentration of product should increase instead of decrease. Hence, 1 should represent product. Therefore, that leaves us with the answers being either C or D. And over time, the concentration of substrate decreases, as when an enzyme catalyzed reaction takes place, the substrate decreases and product increases. Therefore, only component 3 shows an overall decrease with time. Hence, component 3 should be substrate. That leaves us with the answer being D. Question 15. Which statement suggests that the membrane protein is involved in active transport? A. It allows movement of molecules across the membrane if concentration differences exist. B. It can only function if mitochondria are supplied with sufficient oxygen. C. It has a tertiary structure with a binding site with a specific shape. D. It is found in the cell surface membranes and the mitochondrial membranes. Now, in active transport, mitochondria is involved, as mitochondria releases energy by the form of ATP. Therefore, mitochondria should also be involved in active transport. And the only statement that involves the function of mitochondria is statement B. Therefore, the answer should be B. It can only function if mitochondria are supplied with sufficient oxygen, as without oxygen the mitochondria is unable to supply ATP. Therefore, it cannot provide energy for active transport. Hence, the answer is B. Question 16. Cell surface receptors used in cell signaling are usually proteins that extend from one side of the membrane to the other side of the membrane. That means it occupies the whole membrane. What is the role of the extracellular part of the protein? A. Binding the signaling molecule. B. Functioning as an enzyme. C. Increasing the solubility of the receptor. D. Transmitting the signal from the target cell. Cell surface receptors are usually present on the outside surface of the membrane. Hence, their main role is to bind with the cell signaling molecule, as they are receptors. And receptors, their function is to bind with the cell signaling molecule. Hence, the answer should be A, binding the signaling molecule. Question 17. Which substances can pass directly through cell surface membranes and do not use a carrier protein or channel protein? Over here, they're just talking about substances that can pass through by diffusion, and substances which can pass through the cell surface membrane or the nonpolar fatty acid tails of the cell surface membrane are only small and nonpolar substances, as the nonpolar fatty acid tails do not allow the movement of polar as well as large molecules. Hence, if we consider options 1, 2 and 3, 1 is obviously polar. K plus and Cl minus, they are ions, and ions are charged. Since they are charged, they are polar. Hence, substance 1 is obviously incorrect. If we consider between 2 and 3, 3 is glucose, and glucose is large to pass through the cell surface membrane. Hence, the answer is D, 2 only. Only carbon dioxide can pass through the cell surface membrane, as it is nonpolar as well as a small molecule. Question 18. A student put a layer of plant epidermal cells on a microscope slide. The student put a drop of potassium nitrate solution on the layer of cells and observed that the cell surface membrane of many of the cells had separated from the cell wall. The cytoplasm and cell contents had shrunk. What explains these observations? The observations that the student has made describes plasmolysis, as water leaves the cell and the cells have shrunk, as well as the cell surface membrane has separated from the cell wall. So that describes the process of plasmolysis. Therefore, the direction of net water movement should be out of the cells, that is from cells to solution. Hence, considering between options A and B, we should look at the water potential of cells at start and water potential of solution at start. Now, a key thing to remember is more negative the number, lower is the water potential. And since the water potential in cells is initially higher than the surrounding solution, water potential in cells should have a lower negative number than the water potential of solution at start. That leaves us with the answer being A. As in A, the water potential of cells at start is minus 100, which is less than minus 500. Therefore, the answer is A, as water movement takes place from cell to the solution. Therefore, cell should have a higher water potential, and higher water potential is associated with a lower negative number. Question 19. The enzyme telomerase prevents loss of telomeres after many mitotic cell cycles. Which cell transcribe a high concentration of telomerase? 1. Neutrophils, 2. Mature red blood cells, 3. Activated memory T lymphocytes. Cells which undergo multiple cell divisions by mitosis are those that have a high concentration of telomerase, and that is done only by 3. Memory cells divide by mitosis to form plasma cells and more memory cells when they come in contact with a foreign substance or antigen. Hence, the answer should be D, 3 only. As neutrophils and mature red blood cells, they do not undergo any mitosis. Question 20. At which stage of mitosis do these events occur? Centromeres separate, chromosomes become shorter and thicker. Now, centromere separation occurs during the anaphase, and chromosomes become shorter and thicker during the prophase, whereas the reverse occurs in telophase. Hence, the answer is B. Question 21. Which components could be found in nucleotides of rRNA? 1. Any one of two different nitrogenous bases with a double ring structure. 2. Any one of two different purine bases. 3. Any one of three different nitrogenous bases with a single ring structure. 4. Any one of three different pyrimidine bases. Now, statement 4 is obviously incorrect. 3 different pyrimidine bases do not occur in rRNA. RNA has only 2 different pyrimidine bases, that is uracil and cytosine, as rRNA does not have any thymine. Instead, they have uracil. Therefore, only 2 different pyrimidine bases in RNA. Hence, statement 4 is incorrect. Therefore, that leaves us with the answer being B, 1 and 2 only, as all the others have statement 4 in it. Question 22. How many statements are true for semi-conservative replication of DNA in a prokaryotic cell? 1. The process takes place in the cytoplasm. 2. An adenine nucleotide will line up against a uracil on the template strand. 3. Each new DNA molecule will contain one strand from the parent molecule. 4. If the parent molecule contains 40% guanine nucleotides, each new DNA molecule will contain 20% guanine nucleotides. Now over here they're talking about semi-conservative replication of DNA, and DNA replication does occur in the cytoplasm as prokaryotic cells have no nucleus. Therefore statement 1 is correct. Statement 2, an adenine nucleotide will line up against uracil that is only in RNA. Over here they are talking about DNA replication, hence statement 2 is incorrect. Statement 3, each new DNA molecule will contain one strand from the parent molecule. That is correct, as in DNA replication the original strand will uncoil, leading to each strand acting as the template strand or the parental strand. Statement 4, if the parent molecule contains 40% guanine, then each new molecule will contain 20% guanine. That is also incorrect, as in DNA replication if the original had 40% guanine, then the new one will also have 40% guanine. Hence the answer is B. Only 2 statements are true. Question 23. Sickle cell anemia is caused by mutation in an allele of the gene that codes for the beta globin polypeptide of hemoglobin. The diagram shows a sequence of bases in a small section of the coding strand of DNA for both the normal and sickle cell beta globin alleles. How will the mutation in the allele result in the production of an altered version of the beta globin polypeptide? Now as you can see over here, I have underlined the 3 bases which have changed from the normal to the sickle cell beta globin allele, and if you go to the options A, B, C and D, a tRNA molecule with the anticodon GUG will hydrogen bond to the altered codon on mRNA. Yes, that is correct, because if you carry out the process of translation on this mRNA altered codon given, you will get the anticodon GUG. Hence answer A is correct. If you look at answer B, all the amino acids coded for after the mutation will differ. Not all the amino acids will be different, but only the amino acid which has changed due to the altered codon. Answer C, mRNA transcribed from the sickle cell allele will contain the codon CAC instead of the codon CTC. There cannot be codons which have T in it, as mRNA codons can only contain A, U, G and C. T is only present in DNA and not in RNA. Hence, answer C is obviously incorrect. Answer D, the ribosome will be unable to continue translation after the altered codon. You cannot be sure about this because the codon, even though it is altered, it doesn't code for a stop codon. Hence translation will continue. Therefore the answer is A. Question 24. The diagram shows some tissue types in plant organs. Which row identifies the tissue types? They have given the organs as stem, root and leaf, and I have labeled over here which one is which. As you can see over here, 1 represents nothing but the phloem, 2 represents also the phloem, 3 represents the phloem and 4 represents the xylem. Hence the answer is A. Question 25. Which row correctly describes the adaptations of companion cells, phloem sieve tube elements and xylem vessel elements for their roles? If you look at A, companion cell, a thin layer of cytoplasm to reduce resistance to flow, that is obviously incorrect. If you go to answer B, empty cells allowing water to flow freely. For companion cell, that is incorrect, as companion cells are not empty cells. Let's look at answer C. Many plasmodesmata for communication between cells. Yes, companion cells do have. A thin layer of cytoplasm to reduce resistance to flow. Yes, phloem sieve tube elements do have a thin layer of cytoplasm. Xylem vessel element, empty cells allowing water to flow freely. Yes, xylem vessel elements are dead cells, hence empty cells, and they do allow water to flow freely. Therefore the answer must be C. Let's look at answer D. Strong lignified cell walls for supporting the plant. That is an incorrect statement for companion cell, as strong lignified cell walls should come under xylem vessel element. Question 26. Irrigating crop plants with water containing low concentrations of salt causes an increase in the concentration of salt in the soil. What explains why the increase in salt concentration could eventually kill the crop? Now salt is added to the soil, causing a change in water potential in the soil than in the roots. And as you can see, since salt is added to the soil, there will be a water potential gradient caused, eventually leading to water moving out of the cells, as it is killing the crop. If it is killing the crop, that means the crop is losing water, therefore water will move out of the roots. Hence we should consider between answers A and C. If you look at answers A and C, answer A talks about water potential in roots and answer C talks about water potential in soil. Since they are talking about salt being added to soil, we should consider the water potential in soil than in roots. Salt in soil lowers the water potential, leading to water moving out of the plant. Hence the answer should be C. Question 27. The diagram shows a pathway of water across root cells with a xylem. Which cell is in the endodermis? Endodermis prevents the apoplast pathway or the cell wall pathway due to suberin deposits. Therefore, cell C is in the endodermis. Question 28. Which row shows the cause of mass flow in the phloem and the direction of movement of phloem sap by mass flow? Mass flow in phloem is caused by hydrostatic pressure gradient, and the flow is from the source to the sink. Therefore, the only answer that shows that is answer B. Cause of mass flow, hydrostatic pressure gradient. Direction of movement of phloem sap by mass flow, from source to sink. Hence, the answer is B. Question 29. Which statement about oxygen combining with hemoglobin is correct? A. All oxygen molecules which combine stop the hemoglobin molecule changing shape. That is incorrect, as when an oxygen molecule combines, each combination will cause the hemoglobin molecule to change to favor the next oxygen molecule to bind with it. Therefore, A is incorrect. B. 4 oxygen molecules can combine with each heme group. That is incorrect, as 4 combines with 4 heme groups due to 1 combining with 1 heme group. Hence, answer B is incorrect. Answer C. The first oxygen molecule to combine does not affect the shape. That is also incorrect. As I said, when oxygen molecule combines, it leads to change in shape to make it easier for the next oxygen molecule to combine. Answer D. The second oxygen molecule to combine makes it easier for the third to combine. Yes, that is correct. Hence, the answer is D. Question 30. The diagram shows the Bow effect. What causes the shift from X to Y? When curve shifts to the right, it is due to increased partial pressure of carbon dioxide. And since they are asking from X to Y, the curve shifts to the right. Therefore, if you look at the options, only option C and D talks about increased concentration of carbon dioxide. Therefore, the answer must be either C or D. And what is the difference between C or D? And what is the difference between C and D? In C, there is a mention of high pH. In D, there is a mention of low pH. Remember that when carbon dioxide increases, more carbon dioxide reacts with water in the presence of carbonic anhydrase, which is the enzyme, to form carbonic acid. When the acid is formed, it breaks down to form hydrogen ions and hydrogen carbonate ions. And when H plus ions increases, the pH decreases. And when pH decreases, that means low pH, as pH is a measure of hydrogen ions. Hence, increase in carbon dioxide concentration leads to decrease in pH, or either a low pH. Therefore, the answer is D. Question 31. Which plant diagram represents the tissues in a major vein? First of all, if you recall the structure of a vein, veins have a larger lumen than the arteries. Therefore, it should be either C or D. And both veins and arteries, they have 3 layers known as tunica externa, tunica media and tunica intima. And only D shows 3 layers. Therefore, the answer should be D. Question 32. Ventricular septal defect is a heart defect that people can have from birth. People with ventricular septal defect have a hole in the wall known as a septum that separates the left and right ventricles. What could happen in a person with VSD? 1. Blood will leak through the hole mostly from right to left. Statement 1 is obviously incorrect, as the left ventricle is more thicker compared to the right, hence there will be more pressure from the left side than from the right, leading to blood leaking from left to right instead of right to left. Therefore, answer 1 is incorrect. And since A, B and C have 1 in their answers, that leaves us with D being the only answer that is correct. 2 and 3 only. Question 33. The diagram shows the pressure changes in some structures of the right side of the heart during the cardiac cycle. Which structure is represented by X? If they haven't mentioned the right side, then X, since it is usually at a higher pressure from the start compared to others, it is either the pulmonary artery or aorta. And because they have mentioned right side in bold, X should be the pulmonary artery. Therefore, the answer is A, pulmonary artery. Question 34. The diagram shows a section through a type of epithelium. Where is this type of epithelium found in the respiratory system? Now, this is a diagram of a ciliated epithelium which has ciliated cells and goblet cells. Now, if you go through the answers, trachea, bronchus and all bronchioles, ciliated cells are present in all, whereas goblet cell is not present in all bronchioles. Therefore, since the diagram shows both ciliated and goblet cell, It is not present in all bronchioles but only in trachea and bronchus. Hence the answer is B. Question 35. In chronic obstructive pulmonary disease, airflow through the airways is reduced. Which statements explain the reduced airflow? 1. More mucus is secreted into the airways. Yes, that could explain the reduced airflow as it leads to constriction or narrowing of the airways. 2. Airways and alveoli lose their elasticity. Yes, it affects the stretch and recoil function of the alveoli, hence reduced airflow. 3. Many of the alveoli are destroyed. Yes, that could also result in reduced airflow due to reduced exchange of gases. 4. Carbon monoxide binds irreversibly to hemoglobin. Now, carbon monoxide binding has nothing to do with reduced airflow as it doesn't lead to any narrowing of airways. Hence, answers which has 4 in it is obviously incorrect, and that leaves us with the answer being B, 1, 2 and 3 only. Question 36. Outbreaks of cholera commonly occur in refugee camps that are set up after a major natural disaster such as earthquakes. The list shows some measures that can be taken to limit the spread of cholera in the refugee camps. Which features of these control measures involve an economic factor? If you look at 1, treating all drinking water supplies with a high concentration of chlorine, yes, that does involve an economic factor due to the cost involved in supplying high concentrations of chlorine to all drinking water supplies. Therefore, since only A and B has 1, the correct answer must be between A and B. If we go to measure 2, setting up an emergency treatment center to isolate cases of cholera and treat them with antibiotics. Setting up an emergency treatment again involves a cost factor in it, therefore it again involves an economic factor. Since measure 1 and measure 2 certainly involve an economic factor, only answer with 1 and 2 is A, hence the answer should be A. Question 37. Which use of antibiotics help to reduce the spread of resistance in bacteria? Answer A. Using high concentrations of the antibiotic to kill all the bacteria. No matter the concentrations, if the antibiotic develops some resistance, it could still spread the resistance, no matter how much concentration of antibiotic is used. Therefore, answer A is incorrect as it doesn't help to reduce. Answer B. Giving routine preventative antibiotics to people who are having an operation. A key thing you should note about is that antibiotics shouldn't be given as preventative medicine but rather as a cure. Hence, answer B is also not going to help in order to reduce the spread. Answer C. Regularly changing the type of antibiotic used to treat particular bacterial infections. Yes, as even if it develops resistance to one kind of antibiotic, it won't develop resistance to the other antibiotic. Hence, it will eventually kill the antibiotic, leading to less resistance. Answer C should be the correct answer. If we go to answer D, giving antibiotics to treat low-level infection caused by a bacterium, we normally do not use antibiotics to treat low-level infection, but rather very high-level infections. Hence, the answer which helps reduce the spread of resistance in bacteria is C. Question 38. The graph shows amount of antibody produced in response to an antigen. From the graph, which statement is correct? A. It takes 25 days to achieve active immunity. If you have selected this as an answer, it is wrong, as from the graph you cannot say exactly it takes 25 days because according to the graph it shows before 25 days to achieve active immunity, and active immunity occurs after the first peak. Hence, answer A is incorrect. B. Memory cells for this antigen are present in the body within 20 days. Yes, we can conclude this statement being correct, as within 20 days, due to exposure to the same antigen, the concentration of antibody rapidly rises. Hence, we can conclude the fact that memory cells are present. Therefore, the answer should be B. If we go to our answers C and D. C. T helper lymphocytes are activated on day 12. That we cannot say from the graph. And D. The second exposure occurred on day 25. That again is not correct, as 25 lies between 20 and 30. However, the second peak starts before 25. Hence, the answer should be B. Question 39. Which sequence summarizes the hybridoma method for the production of monoclonal antibodies? Answers A and B involve injecting antibodies, and that is obviously incorrect, as the aim is to get antibodies instead of injecting mammals with antibodies. In order to get the specific antibody we require, we have to first inject the mammal with the antigen, and that leaves us with answer C and D. If we go to answer C and D, testing and large scale culture should be done at the very end, hence should be answer D. If we recall the hybridoma method, it starts with injecting the mammal with antigens and then collecting the B cells or B lymphocytes from the mammal. Using the collected B cells, we fuse it with the cancer cells in order to allow the cells to divide repeatedly. Then we test for the antibody produced, as B cells produce antibodies. After testing, we go for the large scale culture, hence the answer should be D. Question 40. The vaccination program for smallpox was a medical success story which resulted in the World Health Organization finally declaring a world free from smallpox in 1980. Which statement correctly identifies the reasons for the success of the smallpox vaccination program? The success was mainly due to the smallpox virus not infecting animals, and also because the smallpox virus was stable and it did not require any booster vaccinations. Therefore, the answer is C. The virus was stable and not prone to mutations. If we go to answer A, the virus also infected animals, that is wrong because it did not infect animals and that was why it was easy to stop the transmission cycle. Answer B, the virus remained in the body following infection. That is also incorrect as smallpox virus did not form any reservoir inside the bodies. Therefore, answer B is also incorrect. If you go to answer D, the virus was unstable, that is again incorrect as the virus was actually stable. Hence the answer is C. And there goes our 40 questions. Interested in answering more papers? Then do click on the next video. Subscribe if you haven't already so you can receive more contexts almost every day. Thank you and stay tuned for more.